Hello and welcome to Nikolay's genetics lessons, and today we are going to talk about gel electrophoresis, and here's another case study. And the question, how to count allele polymorphism from the gel picture. And the question itself, during DNA separation by page, we get one or more than one band for each DNA sample loaded on the gel, but in the case of agarose gel electrophoresis, we get single band or no band for each DNA sample. What is the reason behind this? Here on the left side, that is 100 base pairs DNA ladder. Another question is how to count alleles from this picture. First of all, PAGE, this acronym stands for the Polyacrylamide Gel electrophoresis. And what is the difference between PAGE and Agarose Gel electrophoresis? These are both commonly used techniques for separating and analyzing nucleic acids, DNA or RNA or proteins based on their size. However, they differ in several aspects, including the matrix material, resolving power, and applications. And first of all, matrix material. In agarose gel, agarose is polysaccharide derived from the seaweed, and it is used to create the gel matrix. In agarose gel electrophoresis, agarose gels have larger pore sizes and are suitable for separating larger nucleic acids, such as genomic DNA and larger RNA molecules. On the other hand, polyacrylamide gel is a synthetic polymer, which is used to create the gel matrix in PAGE. Polyacrylamide gels have smaller pore sizes and are suitable for separating smaller nucleic acids such as PCR products, oligonucleotides, and proteins. So polyacrylamide gels can separate bands with a difference even one base pairs. But agarose gels doesn't have this power of separation even if the difference would be 3 base pairs. You would see this as one band, but on the polyacrylamide gel you would see the difference. So this is explanation why when you use PAGE you see 2 bands, and when you use agarose gel you see only one band. But I also want to bring to your attention, for example, take a look. We have a sequence, let's say A, T, C, G, A. So we only, of course, can distinguish between 2 alleles of the different size. For example, if we have insertion here, another C here, so we are going to get a new allele, which would be bigger by 1 nucleotide. Or we also may have, say, situation when we have a deletion. Again, in this case, we are going to have a band which are going to be smaller by 1 nucleotide. So this is going to be another variant of the allele. And of course, there can be different size insertions and deletions. So this is how we can distinguish between different alleles. But if, for example, instead of guanine here, we are going to get, for example, cytosine. In this case, this is going to be 1 nucleotide replaced with another nucleotide, and on the gel electrophoresis, we wouldn't be able to distinguish because the size of the allele is going to be the same, though it's going to be different allele because it's going to be different sequence. But using gel electrophoresis, we only can separate alleles based on their size and not on their sequence. And polyacrylamide gel electrophoresis have higher resolution power. That's why, again, we can see 2 bands. When we're using agarose gel electrophoresis, we may see only 1 band. And this is all for today. Subscribe and see you in the next video. Goodbye.